Test. Also coming.
Congratulations to you. You should feel so privileged.
Yeah. Good morning.
Good morning.
Okay, Jonathan.
All right. So, right on time. Welcome to the last day, last session. I'm happy to introduce Dr. Jörn Rickert, founder and CEO. Am I correct? Cortec? Yes. A company that works with software and hardware for communicating acutely and chronically with the CNS and the VNS. So,
Yeah.
I'll let you go from there.
Yeah, thanks, Jonathan. Good morning. It's an honor to be the last speaker here. I hope you still have some energy left. Few words to myself. Yeah, I'm born 1975. I raised these three kids in parallel to the building of a company, so that's possible to do this in parallel. That's also an important message. Before I continue and go on, I would like to know a little bit about you, just to see what kind of background you have, and so maybe who is has an engineering background? Wow, that's like a large part. Who's kind of a natural science background, like biology or physics or so? Okay, 3. And how about medical? Oh, also quite okay. Good. And who's already on postdoc? Who's doing PhD? Majority. And just being normal students still? No one? 1. Okay, 2. Great. So what I want to encourage you, please, is to engage me in discussion. Yeah? I don't, if I'm just talking for 3 hours, then I consider to have failed. So I really would appreciate if you ask questions, if we discuss, and because 3 hours is a long time, and I cannot talk 3 hours. I have maybe 100 slides, but we can rush through them or skip them and discuss. And please keep that in mind. A little bit more to my background. So I have a background in biology. Studied biology with a focus on evolution, genetics, and biochemistry, and then did a PhD in neuroscience, where I did mainly do data analysis of cortical signals. And then I had some additional qualifications, like training on how to do patterns and how to write a business plan and a little bit of economy things, but I never did an MBA or something, and I would dare to say it's not really that necessary. It's mainly vocabulary that I'm still lacking when talking to financial persons or so, that, okay, is this the right wording for this number in my balance sheet or so? But there are people who can help with that, and so I think there are other things that are more important. And if you want to start a company, then that in addition you think you need an MBA. I mean, it's certainly helpful, and basically you speak the same language of the financial people. But yeah, I didn't really miss it, or seem to have missed it. So then, after my studying, I did a little bit of postdoc, also in this field of brain-computer interfacing and data analysis, and we founded an interdisciplinary initiative down in Freiburg in 2004, 2005, Brain-Machine Interface Initiative, where we had physicians, engineers, and pure neuroscientists like myself and others. And we were lucky in 2007 and got a relatively large grant from a new program of the government that was like 2.5 million, for especially meant for preparing to spin out a company from the university. And there were other funds at that time going into brain-computer interfacing and neurotechnology. But I was then a project manager of this specific grant and used this money to prepare the company. So that was a good situation. Although it's probably still not that much if you compare it to the grants here in the US, and where there's sometimes grants like 30 million to prepare neurotechnology. Everything is a number or an order of magnitude smaller in old Europe. But I could say I was still pretty blessed to start this. And then since 2010, we founded Cortec on paper. 2011, we became operative. And since then, I'm CEO of this company. The talk here or lecture is, we have 3 sessions. First session I'll call the scientific background. I'll just go through what I did in science and what were like the basic motivation that has come from research to spin out the company. Then see, okay, what was the situation when I started the company? And then in the final part, where are we today? Okay, first part, scientific motivation of the company. And there's also special thanks to these guys who were essential in our research and also other research that led to our research. I was working with the motor cortex. And I have a little introduction to the motor cortex. So you all know it's very important in the role of execution of voluntary movements, like when you see something and want to grab something. And the specialty of the motor cortex is you have these. It receives signals from the other cortex areas, and then it has this direct connection to the spinal cord. That's something that is maybe an important foundation, but on the other hand, it has a lot of other outputs going to other cortex areas, going to the brainstem, to the basal ganglia. It has other inputs, receiving sensory feedback from the spinal cord, and then from the cerebellum via the thalamus, and from the basal ganglia. And in the end, you have a very complex network with different types of cells and everything. And the strange thing is, despite this complexity, somehow, what was the basis when also we started our research was when you stick an electrode into this complex network and measure spiking activity, it's very nicely correlated with movement parameters. Force, the first one by Eberhard Fetz, then movement direction, but position, acceleration, all these parameters. And these findings led to the idea of brain-computer interfaces, or more precisely in this one-way direction, as it's meant before Elon Musk has been meant, is the idea of the neural motor prosthesis. Drawing here from Eb Fetz. So goal was to replace the normal pathway, taking these signals, and instead of having the spinal cord process them, use some clever decoding algorithm and then drive a prosthesis, or alternatively, even maybe one's own muscles. And the fundamental basis, which wasn't that large back in the end of the 1990s, there was the population vector from Georgopoulos. It's who knows what this is? Not too many. So that was like a very common knowledge. So here's the spiking activity of a neuron from the motor cortex, and each row here is a single trial where the monkey made a movement to one of these 8 directions. So it's all about finding out movement direction. The monkey did movements, you find spikes, and you see this neuron fires much more to this direction as opposed to that direction, which is actually shows a decrease in activity. So the population vector now would say you record from your population of neurons, and each neuron has a preferred direction, and this is the direction where the activity of the neuron is highest. Now, if you take the activity from all neurons and always take a line that is pointing into the preferred direction of the neuron, then you can, from the average of all these neurons, reconstruct the activity, or the movement direction, which is actually a self-fulfilling prophecy if you think it through, by the way it's designed. But it made clear that from populations you somehow can represent movement. Now, in that time, there were a number of trials going on with more technology and more computing power, looking at whether you can use online decoding or single trial decoding, which would be needed for brain-computer interfaces. You cannot average. You need to do this from a single trial. And this was the field we were working on, but we were not looking into single units. We were looking into local field potentials by that time. So that's also an electrode sticking in the motor cortex. You can record single units from the electrode, or you just take the local field potential, so just a voltage measured at the tip of this electrode. And so here we see a single trial. Not much to see. Another trial. But if you look at a lot of trials, you see that not only is there movement-dependent change in the average activity in the single unit, but also the local field potentials here showed movement direction dependent activity, or you could say tuning. So you could take a point in time, look at the spike activity or look at the local field potential, and you would find a nice tuning curve. Also nice cosine shaped tuning curves in the local field potential, and this was something new that was exciting for us at that time. And we were comparing like how well can you decode movement direction from the local field potential as opposed to decoding from spike activity. And here in this population, in fact, the LFPs were better than the spikes, on average at least, a little bit. There were some exceptionally good in spikes, but on average the LFP was pretty good. Ipsilaterally, even a little bit better. And we worked also with populations then, and in the population the LFP was again slightly superior than the spikes sorted from these electrodes, but roughly similar. And if you combine LFP and spikes, you got the best decoding performance. So that was something we've been working on in Freiburg, and other groups were working on similar things in parallel. And what we then did next was not only look at the time series of the LFP, but also looked at the spectrum. So the time resolved spectral activity with a short time Fourier analysis. So here is like the grand average data of the frequency distribution over time, and there's not that much to see. Time zero being movement onset, again of a sender out task. But if you normalize this and always look what is happening to the baseline before movement, then it looks already more interesting. You see there is something happening. mainly there are, in the low frequencies, you have an increase in power during movement, and there could be maybe another band that's not as low. There's also an increase with a slightly different dynamics. There is a decrease in the beta band, and then you have the higher frequency or gamma band activity where you find an increase. And then again, next we looked, okay, now is there movement-related activity in these different bands? And so here is an example of a local field potential. Here's the time signal, here's the average spectrum. And you can see that depending on the movement direction, there are differences.
Excuse me?
Yes.
I have a question. Can you come back, please, to the previous slide? Do you have any interpretation for the previous to the zero time to the waves? The change previous?
You mean the increase here?
Yeah.
Or the changes?
So in your right plot, you have a change of behavior before time zero, right? Yep.
Well, yeah, I mean, okay, of course it's known that there is activity, of course, relevant to planning, so that there is something happening, needs to be happening before the movement.
Right, right.
I mean, there is
So
clearly
my
a
question
delay.
is,
and then, of course, you also have an averaging effect because you somehow have to average time data. So both together make it, yeah, or it's clear that there needs to happen something already before. And yeah, it depends a little bit on the frequency band.
Okay.
And
Thank you.
what. So maybe this is not new to anyone here, or hope anyone who's working with signals now looks at different frequency spectra and looks at different signal components getting from different frequency spectra. But okay, at this time, for these signals, it was new. And we looked at, again, tuning curves now for different frequency bands. We found nice tuning curve in the low frequency, in the middle to low, no tuning in the beta band, and again tuning in the gamma band. And it could always nicely align also to cosine tuning curves. So in the end, looking at the signal to noise ratio, it's like an equivalent measure to decoding power, representing how well this frequency band is tuned to movement direction. Looking at contralateral LFPs and ipsilateral LFPs, we found that contralaterally the gamma band shows a high frequency. Yeah, the highest signal to noise ratio, or the best representation of movement direction, not so much ipsilaterally. Low frequencies are very similar on both sides of the brain, and you get nice information also on the ipsilateral side. And the beta band essentially didn't show any movement-specific activity, so this seems to be more generically related to onset of movement or planning of movement. The actual starting point of Cortec then was when we have been working with humans. So this is an epilepsy patient undergoing pre-surgical diagnostics. He had this large grid implanted over the surface of the brain. I guess you heard a number of lectures mentioning these patients who are very interesting for research, because they're essentially sitting in bed and waiting for seizures, having electrodes implanted, and thus allow, if they are willing, to do some kind of experiments. because they're implanted anyhow. So it's the very rare case where you have a human subject being implanted and can do something. And so we did just here like a movement task with this manipulandum, and then you see the brain activity flickering here. And so we reconstructed the single trial, taken from the single trial brain activity, the movement, and so you see the movement he's been performing here actually in green, and the movement we decoded in red, and we were surprised that it worked quite well given the few time you have with these patients. So there's no real training of the patient happening. The electrodes are, really large. Over the motor cortex and the areas here of the arm, there are maybe 1 or 2 electrodes, so really precisely over there. So we thought, oh, this is a good perspective. If you optimize the electrode, if you go for chronic training, we thought we should be able to get much better than this. And we did some more experiments. We looked at grasps and whether you can decode different types of grasps from the ECOG, and looked at how well the different frequency components, again, low, middle, high frequency, just a time-resolved local field potential, and how well you can decode the grasp. And this looked similar in quality again to the papers you had from the spiking activity. And we also did an experiment where we wanted to see how stable or how robust is the decoding if you are in real life where you have different conditions. Typically all the studies are done like, monkey or human sits upright, does this, fully concentrated on the task. But in the real world, you do movements in parallel to talking. You may lie or sit. You may watch TV. You might do something with the other hand. And so we did an experiment where the subject was moving a cursor left or right, a joystick in fact, but under all these different conditions. And we wanted to see, can we still decode the movement when you train the decoder with data from one or more conditions, but then use it in another conditions, because that would mean what will happen in real life with these devices. And so here you see the again, as an example activity of an LFP. And I think this is low and this is high frequency, movement to the left in black and movement to the right in red across the 6 conditions. And you see fairly consistent tuning curves. And then we also did a decoding time series, or here using the gamma activity. And you see the decoding accuracy. It is better if you use only the data from the same condition, that's clear. But we could generalize. So the black curve shows the decoding accuracy if you train it with data from other conditions, and the condition you're actually decoding is not included in the training data. So in essence, all these studies on the local field potentials and on the ECG made us optimistic that you might use these type of signals also for brain-computer interfaces. So single cells are obviously a source for brain-computer interfaces. There have been all these experiments with the Utah array showing very nice results from Andy Schwartz and John Donahue that you can use them. On the other hand, a lot of work has been done with the EEG. There has been a famous paper with the Hans Salzmann here, like the first really heavily paralyzed ALS patient spelling a letter to Niels Bierbaumer. I think this was like in 1990, I don't know, Jonathan, do you remember, like 8 or something? So, but it wasn't clear whether you could also use these in-between signals, local field or ECOG, and I think with the research that we've been contributing to, one would say yes, you could also use these signals. And on the question like, what kind of signals you should use best for a brain-computer interface, we thought the LFP taken from electrocortical gram and so electrodes located on the surface of the brain might be a very good compromise, because you want to be here in the green spot, so you want to have a lot of information. You don't want to be too much invasive, and you want to have a high stability of the signal. You don't want to, have signals who vary too much or get lost or have to recalibrate it, or signals like the EEG who are very easily susceptible to artifacts. So, and we thought if we do a micro ECOG on the surface of the brain, that would be a good candidate for a brain-computer interface. So, from this research, then we were motivated to develop this, but it was also clear you cannot do this at the university. To really develop such a brain-computer interface and in order just to allow proper research, you would need something that can be used chronically. And this means you need a wireless device where there are no cables sticking out of the head, and you need a higher resolution device. And this was not there, so we had a long discussion. Do we really want to do this invasively or not try to go for an EEG-based system? But we decided, yeah, okay, let's try for an invasive system, and then that will be the next chapter now, preparing a startup company. I don't know if we should do a short coffee break or move on. I think everybody has a coffee. I don't know, but
We're only going for half an hour, so let's go.
yeah, we'll continue. But anybody who wants a coffee, then just in between get a coffee. Okay, so I'll not come back to science anymore. So that and these are the original slides we had in our early on presentation. So in the end, you might also see an evolution of the maybe marketing quality of the slides from start to end. But this was like the slides we were doing the first pitches here. So that was our plan. We wanted to develop a 3-component implant system we called BrainCon at that time, consisting of an electrode, the so described or motivated high-resolution ECOG electrode. Then we would need a wireless interface communicating through the skin. And then we would need a decoder sitting on the outside that would allow for control of assistive technology, brain-computer interfaces, and probably more applications. And so here's a little bit the timeline from, and don't be shocked, it might be faster also with more money. But like we founded this initiative in 2005, and then finally Cortec was spun out here and become operative here. In fact, the first business plan, I got a little grant after my PhD, just a year to write a business plan, and we filed some first patents back in 2006. And then 2007 was what I mentioned. We were lucky to get this specific grant to prepare a company. We got some more grants and more patents, and then came the phase, I'll come back to that later, where I started raising the initial funding, and we did also IP transfer then from all the university IP into the company. University got some shares in exchange. And in parallel, my personal line, science and research, went down more and more. At the beginning, I was more still doing in science, a little bit of planning, but at the end it overtook, and since then it's zero here. The constellation where we have prepared Cortec from was this so-called brain-machine interface initiative that was founded in parallel to this timeline that you see. And so we had 3 parts. So there were like I was part of this Bernstein Center for Computational Neuroscience. So there were like hardcore neuroscientists and decoders with Art Artson and Carsten Mehring. Then we had a hardware development. So that was like the engineering department of the university with Thomas Stieglitz and Martin Schüttler. We also had a company being part here, part in grants we had. This company Multi-Channel Systems, some of you might know, they're very active in the research field. And then we had the clinics here where we did these studies with the epilepsy patients and also animal trials. And we had the clinics here in Freiburg with Tonio Ball and a group of Andreas Schulze-Bonhage. And we also worked closely with Tübingen, with Niels Bierbaumer, and at that time also with Alireza Garabaghi. Yeah, and so the idea was, okay, to spin out Cortec, which looked like this from the logo at that time, out there and, do medical device commercialization from the research that's done in this initiative. So in the essence, the business model I had in mind at that time was like that Cortec is the transfer center for neurotechnology for this brain-machine interface going on. So you had all these academic groups working on this innovative things, also with a company involved. And in 2006, we also received a Neurotechnology grant, a large one, about 10 million to push this type of technology in many projects. So Cortec would take the technology and make it into products that are either sold to other companies or brought to the market. So that that was the idea at that time. Again, it was nice for us to benefit from support from this network and the public funding. And okay, so but the plan is, okay, the parts of this technology here for this BrainCon coming from this network, and probably in the future more, but like the core project was to realize this. It was called BrainCon at the start, but meanwhile it's Brain Interchange, and it was the status in 2010 when I started the company, or I was trying to start the company. That's what I want to show now, where we were at that time. And okay, first, so the 3 components. First component, the electrode. So we had a new technology out there in Freiburg for developing ECOG electrodes with a high resolution. In fact, we had 2 technologies. There was the thin film technology using polyimide, and the laser patterning or laser ablation technology using established materials, silicone and precious metals. And we chose this technology with the silicone and precious metals also because for us as a small company, it looked much easier to get approval for something based on established materials as opposed to getting newer materials into the market. So it's just a new manufacturing technology using traditional materials. So that was how the laser looked at that time. It was actually a normal printing laser, normally made to print some texts on whatever labels or so. But it was used to ablate silicone and metals. We could get down to feature sizes around 40 micrometers in platinum and 150 micrometers in silicone. Essentially, the engineers will do the design on the PC, forward it to the computer does the processing, the laser does the processing, and then step by step you generate your electrodes. We did, very importantly, of course, early on cytotoxicity tests, which looked very well. So that was good. So that this laser method didn't seem to produce any cytotoxic effects. We did tests of actual high-resolution electrodes in mini pigs and sheeps at that time. So that's how the sample electrode here looked like. It was like a 1.7 millimeter center-to-center distance of the electrodes. And so we were looking, for example, can you, when you poke the snout of the pig, differentiate different locations? Does it actually make sense to use this high-resolution electrodes? And yes, we found very localized responses on these electrodes as a test. So high resolution, this pig experiment made sense. Then what we did next was to implant chronically in rats at that time. We took a population of 13 rats. We made a very small electrode. You already see one problem in neuroprosthetics. You want to have very small electrodes, but in the end you need to connect it to something, and then what often happens is the connector is much larger than the electrode. So this was a fixation bar cemented into the bone. It also developed this interconnection technology, and we implanted them for around 6 months, did regular measurements and did histology. Afterwards, this video takes a while. It's not that specific. The interesting thing is with the rat that compared to the human, the dura is extremely thin. And from also what we learned is that doing pigs and sheep and rats, and the reactions from the histology look quite different. We didn't do enough statistics to really compare this, but it seems to be a problem in that it's not so easy to transfer results from one species to another. And especially if the dura is so different, and you see here, this is now the dura that's opened. It's very, very thin compared to the human, more leathery dura. We did recordings from the rats running around, and importantly, we looked at the impedances of the electrodes over time. Here's the frequency and the impedance, and the curves are showing the development over time. And the important thing is that after initially impedances are better, then you have a tissue reaction, or most likely due to a tissue reaction. We cannot completely confirm this, but after a week already in the rat, the impedances became stable. Then they remained essentially stable until the end of the trial. And also the signals remained stable. So that was like the situation, what we did with the electrodes. We had some animal testing, some long-term testing, some cytotoxicity testing. We had the technology developed. How did our wireless interface look at that time? Essentially, it looked like this. So it was essentially an electronic circuit here developed by company Multi-Channel Systems. Was designed to record from 32 channels, 2 kilohertz, 16-bit, having an infrared communication of 1 megabit, and it was powered externally by electromagnetic induction, designed for stable energy supply and high data throughput, and yeah, and we were at that time training the chronic implantation in sheep. We were getting sheep heads from the butchery and looking on how to open their skull and put the electrodes in and where to place the wireless interface. But you see, okay, here's obviously no encapsulation. So we have been developing a ceramic metal encapsulation designed for chronic implantation. This was in the end also an already established technology in a certain way, because it was used in one commercial implant, the Actigate implant. That's an implant by a company, Neurodechnologies, initially then acquired by Otto Bock. It was for a drop foot stimulator, so an implant sitting here and in the end stimulating nerves so that in stroke patients automatically you move up your feet. But we, or let's say the engineers at University Freiburg, they refined this technology such that you can have a very high number of feed-throughs on a very little space. So because the existing implants or the existing implant encapsulations are these titanium cans derived from the hard pacemaker technology. With their header, they typically only have 8-channel feed-throughs, or maybe then you have 2 and 16. But with this technology, we could have 300 channels on a thumbnail, and so this was being developed at the university. So here's the manufacturing process, and yeah. But you see already here from the status of this graphics that it wasn't that much developed. You know, this is obviously a cartoon, so it's not really there. Yeah, but so that was in development. And then the other thing was the decoder, or actually the software driving it. At that time, we had a research-based, pretty large and complex software already designed to collect data, filter data, decode data, apply different paradigms. Was working in real time. Was tested experimentally. We had some safety tests with in Europe, it's notified bodies who inspect you, and so we had already some dealing with regulations, more than 100,000 times of code, and so here's one of the experiments we were then doing, trying out this software. So this is a patient, and it's actually a real movie, so he's not moving. His brain activity is moving though, and yeah, he was just playing this very, very simple brain pong. So there were others doing more fancy experiments, but for us this was a proof of our technology. Again, we also had a car racing game where they were playing a racing car game, but it isn't as good as this in a video. Yeah, but that was essentially the status, and of course, then when you want to start a company, you explain all the technology, and that's great. But in the end, that's not what the investors interest that much. They want to see, okay, where do you want to go? Where's the market? And so the target markets we had at the start in 2010 was we had the plan of taking the electrodes first because they were furthest developed, and you could make a standalone product from this, and that we would have our hopefully better or higher resolution electrodes that could then be used in the epilepsy patients or in intraoperative monitoring. We would estimate that the total and intraoperative monitoring is a higher number than the epilepsy. That's around maybe 8,000 patients a year, so a small market, but it's a market that's there. And then for the complete system, we were aiming of brain-computer interfaces and paralysis for computer assistive technology. And we also had a project with University Tübingen where you aimed for stroke rehabilitation, but that was further away. We were also thinking of treatment of post-stroke pain with an ECOG by that time. And essentially the core was here, all the products here would be medical devices made and certified by our company and sold to the clinics. And that was our time plan here that we discussed, with a lot of experts from the field. And even the TÜV, the notified body. So by that time, the market approval of the electrode should be very simple, because it is essentially a product that is on the market. Same materials, so we'll just copy it. In the US, this would be a so-called 510K submission, and in Europe, it would be a literature-based submission where you just show your device is equivalent to what's in the literature. So then in the second year, we would start selling the electrodes. In the third year, we aim to go for clinical trials with a BrainCon system. And then a year later on, we've completed the trials and prepare the market entry of the BrainCon system. And nothing of this has happened. Yeah. Okay. I'll come to what happened, and in the end, we got this year a market approval for the electrode, but not in Europe, but in the US. Yeah. So there were a lot of things happening, and I'll come to the story. But apparently, looking back from standpoint of today, that's a ridiculously ambitious business plan, especially given the funding we were aiming for. And the funny thing is that you develop. I mean, I didn't develop this business plan alone. I mean, I was having advisors supposed to be very experienced, but it's clear it's a kind of optimistic plan, but not an unrealistic plan. So and even the TÜV, which is the certification equivalent to the FDA. So we had discussions with them, and they said, yeah, it's realistic. You can make a literature-based approval, etc. And so yeah, that was the plan. I started out looking for seed financing, and I started this beginning 2010, and in the end completed this in the summer 2011. So I was pitching to investors at some kind of venture capital summits and participated in business plan competitions and what you do. And in the end, the outcome was that I had an interest from 3 companies, or strategic investors, as they are called. And we were invited to 7 presentations at venture capitalists and business angels. And after the due diligence there was one venture capital fund, 1 strategic investor, 1 private venture capital, and 2 business angels who were still positive and wanted to invest. But the strategic investor, we somehow didn't realize. Maybe we scared him away, or he retreated. So in the end, when I started the company, this was what we had. We had 500,000 from a startup venture capital fund. We had a business angels, 150,000, a small side investor, and founders. And you see, so like 800,000. And this was leveraged by public grants. So in Europe, you always need investor money or private money to co-found grants. You never get 100% funding for a company. So I could never start a company only on grants. So this was a kind. Normally you need half. So I leveraged this more or less to the extreme and got 2 million in public grants to start. And that was the starting team back in 2011. So that's me, and then we had a little team moving from university into the company. Here, Marcus, he's a biomedical engineer who then took the task of doing all the regulatory, all the quality management. So it's like well, either you do this or we have no position for you. Okay, then I'll do the quality management. And okay, meanwhile, he's still on board. He likes it. Jörg, also doing in parallel his PhD thesis, was the software developer. Chris here was the electrode guy, the electrode developer. Wolfgang was an engineer who joined also. All these people were on board for a while during our university projects. He came from industry by that time, so he was heading the manufacturing. And Christina, actually an old friend of mine, once looking for some job to complete her, what is it called, doctorate in history, then joined us for marketing and sales. Sophia was also an assistant in manufacturing, and Martin and Thomas Stieglitz had permanent positions at university. They also got some shares because we needed their clean room to do all our operations, but they stayed at university, so it's essentially a small team. I shared some shares with all of them, but yeah, we had like everything, but everything just one person. Yeah, so that was when we started. So and now I think we make a little break for refreshment, and then I'll tell you where we ended up. But you're happy. You're welcome to ask questions, of course. So a lot of time has passed. So what does Cortec do today, and how do we position ourselves? So we took this slogan that we enable communication with the brain and nervous system for the cure of disease. We think there is a lot of potential in realizing the communication with the brain in both ways, reading from the brain and writing onto the brain by electrical stimulation and reading the electrical activity. And we base this on the brain interchange platform technology. That is what came out from our first BrainCon project. And I think that this has a lot of potential, because what more generally, so you have all the electrical devices, they are all functioning by electrical current, and the communication in our body is also working with electricity. So this interface is going to be interesting. And there's so much going on in the research field with the bioelectronic medicine coming up, and more studies going on, that we are optimistic. So what has happened since this seed funding, which took place in September 2011? So the first thing that we did on the technology side is that we ISO certified our development and manufacturing for implantable electrodes and active implants. So I think in the US, and this ISO 13485 is largely equivalent to the, I think it's the 80 CFR 2080 or something, quality management rules from the FDA, which means you need to make sure that you have consistent manufacturing conditions, that you can trace back every single item you build into a device back to the supplier and back to the specific delivery charge that you've been received. Here's a picture from the old clean room. So that's the small end clean room. There's a gray, also controlled room area around it. That were about 80 square meters in total that we had at the university. And we first built the quality management at the university before Cortec, and then did this. So that's a lot of routines, and it's still a bit strange, because when you do install a quality management system, it needs to go all the way to how do you sell to market, how do you make and monitor the products onto the market. Well, you are not selling anything. You're not even producing something that's complete, but still you need to have these routines in it, which in the end means you come up with something you think is conform to the rules, but it is not really working very well. And in fact, we had 4 upgrades of our quality management systems, or maybe even 5, depending on how you count, during this time, in order to really adapt it to the company. And it's something that you think you do it once, but it's a continuous process, taking continuously resources. You remember this first picture of the laser? We added a new laser. The first one was a nanosecond laser. Now we installed a picosecond laser at university, and now we even have another generation, so they were much faster and more accurate. And we did one very important thing also. We decided to include stimulation capability to the implant. When the first device could only record, so it would be good for these brain-computer interfaces. Although also there, I think it would be better to have stimulation included to allow for some feedback and better training. But with stimulation, obviously, you could address many more needs and all the other therapies, from Parkinson to epilepsy, and so much broader application. So that was a strategic decision, and so we thought if we do this, we would have an advantage to what's going on, because many competitors were working only on recording devices. So this is the so-called Vimagine implant that has been developed in Grenoble in France. So also a state-owned entity, actually managed by the inventor of the deep brain stimulation for Parkinson, Benoit Dubuis. No, what was? Anyhow, they could only record, and then there's also the Brown implant had been developed for the Utah array, 100-channel recording implant that has been transferred to the Wyss Center in Geneva for further development. And on the market were the products from Neuropace, you heard yesterday, but also Medtronic has their closed loop device. And so these are essentially devices based on the old technology, so they don't have many channels, and they have very limited recording and computing capabilities. So all the processing is done on the system, so you have limited battery power, so you cannot really use the algorithms you typically use as a researcher. And if you do continuous recording, the batteries deplete quite fast. So we thought having a higher channel wireless system is an advantage, and we think this still today. So the Cortec brain interchange is a system developed to enable personalized neurotherapy right on demand. So it is using established technology. It's very safe for the patient. There's no battery inside. And here's a little video on how this system should work. What we did, and what we also could also frame it, so that the system we have connects the information highways of the human body with the means of artificial intelligence, because you have all this external processing, and we developed all the little puzzle pieces that you saw. I'll come back to that. So, but the basic idea of how this system will operate in human, it's not in human yet, I'll say that. So you have, for example, a continuous recording from an electrode on the surface. It's transmitted wirelessly to the outside. And here it's processed by a handheld device within a few milliseconds. So a very short turnaround time can include information from other signals like EMG or the environment. And then the stimulation demand is computed and sent, for example, to an electrode deep within the brain. And this system then operates continuously, analyzing demand by recording brain signals and other patient parameters, and then delivering stimulation on demand, for example, to have a better treatment of Parkinson. And so essentially that's about closing the loop in neurotherapies. I mean, there's a lot of research thanks to Neuropace and others, but mainly motivated clinicians going on towards closed loop. But still, the majority of all patients implanted, they receive open loop therapies. And that is something which is clearly not natural. And it's to any knowledge not the best of a therapy. Imagine for the heart, like the first implants, the first neurostimulators were pacemakers, and still the most widely used implant. And the first ones were just delivering the same stimulation to the heart. So no matter where you were sitting in your armchair or running up the stairs, you would get the same heart rate. And since a long time already, heart pacemakers are closed loop, so they adapt to demand. But the neurostimulators, they so far don't, and they just started this. And there are already studies from bedside studies for Parkinson demonstrating that if you do it closed loop, it's a more effective treatment. And but the technology to do this is not yet fully there. And that's the niche where we want to step into with our brain interchange technology. So in the end, we continued the development of what I've showed you at the beginning with all the different puzzle pieces that are needed. So from the electrodes, the interface to the nervous system, we're also working on a high channel interconnection. We have this high channel hermetic encapsulation. We developed our own electronics, wireless communication and power supply, and then the external software that is controlling the system. And that's how it looks like today. We have an implantable part, which is essentially different types of electrodes. You connect then hermetically to implanted electronics that are protected by this ceramics encapsulation. And they communicate wirelessly and are powered through the skin with an external device. And the processing is done at this stage still by a laptop computer or any type of computer or tablet. And in the future, this will be included in this external device here that the patient can wear on his arm or on his belt. So that is the strategy that we came to develop, not only a BCI for recording, but a device able to record and stimulate and to enable fast closed-loop cycles. So here's again a picture of the device. So in this version, we have 32 channels that can be used at any point in time, either for recording or for stimulating. And we tested the technology in chronic animal studies. And yeah, we didn't do anything new. The whole system as a whole was designed new, but at least we used established materials. So all the biocompatibility tests we did so far, and it has been a lot, were positive. We established an implantation procedure that's also not new. It's implanted similar to a cochlear implant behind the ear. Yeah, and I'll show you a little video on the animal trials. And I mean, animal trials is also a critical thing. In Germany, we have very, very negative public opinion on animal trials, and the university clinics, they even didn't want to allow us, just having this movie. I mean, and showing this, and it's something I have to add. You have to exclude the animal video part from the live stream. It's good that I remember this because they are also separate action potentials and local field potentials. It got improved twice, decreasing essentially the noise floor. And we can then connect the stimulation input to any of the recording sites, current controlled stimulation with these parameters here, where essentially you can do all the typical stimulation parameters that can also be done with the Medtronic or NeuroPace implants. So this ASIC is now in the current version of the system that we are validating for human use. But essentially the ASIC development, which we are doing with University Ulm, with Maurits Ortmanns, who's a really genius in neurostimulator ASIC circuits, and he's always ahead of us. So he already has a 32-channel combined recording and stimulation ASIC, where you can, on all channels, apply different types of waveforms in parallel, and is also working on a wireless communication protocol with 100 megabits. And the problem is always that you have this new components already available, and then the engineers in your team would like say, yeah, let's put it in the system. And at one point you have to say, no, we need to come out with something, you know, because if we take this and include it, it's another 2 years until we have it with all the testing and development needed. So it's important to in medical device, it's called design freeze. So you have to say, okay, that's the design we take. We go with it and bring it out. And then, okay, maybe we do a next version. But if you continuously improve it, and so you will just never come out with something, at least not something for a human, and even for animals, maybe it will not be very reliable or stable. We worked a lot on the electrode. I mean, the electrode, from the basic principles, the technology has been established, but we established the manufacturing according to medical device regulations, and we extended our technology, which only was meant for ECOG at the beginning, to cuff electrodes, which was something we were really not sure by the time we were approached by researchers and also some startup companies. Some are quite well known. Well, could you give us cuff electrodes for our research, specifically very small cuffs, because there were not many small cuffs on the market, and we thought, oh, this will be a distraction for us on our way, you know, to get approval. And but we did it then using the same technology to make cuff electrodes, and we have much more business going on now with cuff electrodes than with the other electrodes. So in the end, it was a very good decision, but by the time we were really not sure. And yeah, we went for the CE approval as planned. So essentially, we established the manufacturing. We did start all the testing that you need. And so, I mean, we had the electrodes, we could build them, but okay, you need to have a design history file and a full documentation set to get market approval. So we worked on that. And as I told you, the business plan was to get it approved like in the first year. Because it seemed straightforward. I mean, we did pre-test, everything looked good. But so why did it take so long? So the first thing that happened was, of course, things took longer when you do them than you plan. But we came to the point where we did the cleaning validation. Cleaning validation meaning that's also why you need a clean room. You have a stable environment, and you have to demonstrate that the electrodes are reasonably clean, and that's done by sending them to a test laboratory. Then they do an extraction, and they count the number of particles that they find on the electrode. So we took the best and most well-renowned test laboratory in Germany for these types of tests, and started the testing and got inconsistent results and often had very high numbers of particles. The point is we didn't, or how to find out where they are from. So we changed processes in our cleaning machine. We changed from using the university provided purified water. We didn't really trust the university quality management, so we bought bottled purified water for the washing machine. Half a year was gone by doing this. Nothing changed, We were like looking into strange things like, is there an influence of whether you dry the electrodes when the sun is shining outside or not? You start, because essentially you don't know. Then, 9 months had passed and we were starting to get nervous. I was going to Thomas Stieglitz and Martin Schüttler, who were like heading the laboratory with their university positions, and telling them, I don't know, I need one of you to join the company somehow. That's not going like this. We need more expertise there. What we then did was, in the end, we took just a single sheet of silicone, just a single silicone sheet, did all the processing, and at the very end, after all the cleaning, we cut it in 4 pieces and sent it again to the test laboratory, who was regularly visiting us and giving all kinds of recommendations. The result from this was like the first part 1 of the 4, 0 particles, great. Number 2, 100 particles. Well, okay, but still below threshold. Sheet number 3, the part number 3, maybe 2,000 particles. That's already above threshold. But then slice number 4, 150,000 particles. Like, what? How could that be? It's like everything done the same, just cut at the end. So, then we changed the test laboratory. Went to another test laboratory, did the first test, order of magnitude under the threshold that we needed, and never went up again from that time. So, okay, but we lost a year, and a year was like only particles. And so these are things that can just happen. Then in parallel, we got new medical device regulations, which were clear, like we just have a very little time because rules are going to get stricter. You need more clinical data and testing, and but finally we submitted everything to the European well-renowned notified to our notified body, which we have been working on for like 4 years or so, and there were also funny things happening. Like then after Christmas, I was asking our man there what the status is, and got an email, I'm not with the company anymore, and because they were bought, the notified body was bought by another notified body. They tried to integrate it, they failed, so essentially they dissolved, and we were still hoping to get it done, but then we received another problem. We had done all tests with positive results, they were sent in, except for the validation of the transport packaging. So you need to make sure that your sterile product shipped throughout the world remains sterile, so the packaging needs to remain intact. What they do is they wet it and then see, is it still germ-free, so that there can no germs pass through. And this test failed. And we did steam sterilization, normal steam sterilization with a package that was supposed to be used for steam sterilization, and we tried all kinds of other steam sterilized packages we could get from the market, but somehow they didn't pass the test. And again, we didn't know really why, and then you could now fight with the packaging suppliers, but it wouldn't help us. So we had no choice. We changed from steam sterilization to gas sterilization. But if you do this, you change your manufacturing process, because in the end you have another manufacturing process. And this means that you have to repeat all tests, all biocompatibility, all mechanical, all tests, So, so these are things you cannot predict, and they just, happen. And so that was another year, and then we ran into our notified body had dissolved. We went to TÜV Süd, and they they increased the regulatory hurdles. And by the time we had resubmitted to them, we did a lot of improvements. And meanwhile, they told us, well, it's now too late. Now we have the new regulations. Now you need clinical trials in order to get product approval. And yeah, so by that time, it was really shifting towards the US, which we were a little bit ahead then in getting US approval. And yeah, so in this March, and US approval first, we did try with a partner company, but this partner company then dropped out of this project, got sold, and then, okay, we restarted this on our own. So finally, we got FDA clearance in this March, so which is like 8 years from the start, and for a very simple product, because a lot of things happened. But anyhow, we are doing business with our electrodes, so silicone-based, but finer than what is there. We have established a production line, can do all kinds of different designs, and yeah, we are doing this. And well, here are some of the specifications that we can make. So here are some designs. We have different electrode contact materials. We have established coating processes. Contact diameter can go down to 50 micron. Using a multi-layer process, we can get up to 100 contacts on a square centimeter, which probably not really making sense for human applications or chronic human applications. We can modify the mechanical robustness by Parylene C. Yeah, and we're very, very happy with these electrodes. Here's a design example, like a large grid with some small contacts or micro contacts for research. Here's a product where we combine an ECOG with a needle array from the company called Microprobes. Here are designs we make for the peripheral nervous system, where we have like these meander-shaped contacts that are stretchable, and so you can have a very large contact surface, and with very soft electrodes that nicely adapt also to swelling nerves. And we have different types of designs and closing mechanisms. Here's like a buckle and belt that's used a lot in small rodents. That's a tunnel cuff design. Here we also have spiral cuffs, and they are used in large animals and also increasingly in human studies. This shows a little bit the variance in size here. This is this little buckle and belt sling cuff used in rodents, and this is a cuff that has been used in the human arm nerves to enable targeted muscle stimulation. Yeah, and if you open it, you see it is the same technology as used for the grids, but you just shape it into another form. The encapsulation has been validated and fully tested. So here's the implantable electronics, then gets the ceramics on it. It gets sealed here by the soldering process. Here you have the feed-throughs, so then you here solder the electrode contacts and the power supply call, and then afterwards, and that's a very important step, everything is cast into silicone rubber using a specific mold, and for this we have developed a specific vacuum centrifugation process, because this silicone here needs to be absolutely bubble free, so that there will be no corrosion later on because of water coming in. And after some iterations, this encapsulation works extremely well. Here's the manufacturing process that has been developed. And yeah, there is a critical process also where you use a pressure curing, and then in the end you remove it, and then this is here what you get out of it. We even were very happy that we were so robust, although it's a relatively thin ceramics, that we passed this hammer test. That's one standard for active implants coming from the cochleas. And you know the history? Does someone know the history why there's this hammer test and where it's coming from? So it's coming from Australia, and they are playing there a lot of baseball, and they realized that cochlear implants are implanted a lot in kids, and so what was happening is kids playing baseball and being hit by a baseball really hard tended to smash one or the other cochlear implant. So this test is mimicking the impact of a baseball. It's designed like this. Like the hardest possibility, and so we are happy that we were able to pass this test. Most important is the lifetime of the implants. So we did a number of accelerated aging tests, and importantly also another study in sheep where we just took 5 implants, put them in a sheep, and watched them over time, and then left away the so-called desiccants. So you put some water draining powder into implants, and so we did some extrapolation studies, and there should be an expected lifetime that should cover the lifespan of people. So we had the encapsulation, and we also meanwhile have some projects with customers where we use our encapsulation. Then we developed our software and firmware. And I mean the software we had initially at university with this more than 100 code lines, I don't know if we even have it somewhere, so that's pretty much irrespective. So the software is also something that was totally underestimated from the start, because in the end you have so many different levels. You have the firmware, you have all the safety controls, and so I'm not a software engineer, but it's now we have a team of 6 software developers who are working full time on finalizing all this, and it looks well, but it's a big project because you need to do it also under design control and do all the specific testing and documentations also on the software side. We filed for more patents. I think we started with 8 that were transferred from university to Cortec. Currently we have 22. We have 3 trademarks globally. That's like most of the patents are on the electrode technology, where we also have most projects going on. Other aspects of the brain interchange, but also a number of specific applications, BCI or closed loop, where we had some good ideas. We built some IP. We are working a lot with partners still. We have the University Freiburg, There was an excellence cluster, BrainLinks Brain Tools, which is now running out, but there are a lot of collaborations still with the Institute for Microsystems Engineering. We are continuing to develop the technology. We are working with University Ulm to develop ASICs and improved ASICs for our implant. Also with these, we are working on MRI compatibility, which is, everybody wants MRI compatibility, but that's really, really complicated and costing a lot of time and effort. Yeah, so we are still connected. And now let's stop from the technology side and look to what else we do and develop. And that's business development. So essentially, we completely changed our business model. So obviously, the regulatory hurdles went up and up and up. So we are now in the fourth increasement of regulatory hurdles, new regulations. So so it's just extremely costly and also risky to go for a new therapy. You remember the talk from Nevro? So they are now like more than 20 years old. They have a market-approved therapy, but they are not making profit. And okay, I mean, you can maybe be more lucky and it works out better or so, but in the essence, what we have developed is a platform technology with the brain interchange, a platform technology, and we want to use this platform technology. And so how do we want to do this? So we have developed this new technology, this system with all the different components. So we went for a much more modest business model with 2 steps, which I still think has very nice potential. So what we are currently doing is technology business, very simple. We deliver customized electrodes or hermetic encapsulation or other components, and in the future also complete systems where we also get requests. And we build this for typically industry or startups. We include documentation. We know how to do this with all the experience we made. Or for large academic centers who want to develop something for a human application. So simple, like more like a butter and bread type of business. But there is a good demand because there are not so many companies who have developed innovative technology and make them accessible. Because typically companies, when they have a new technology, they want to use it for their own killer application. And so we are very happy with this business and how it develops. The other side is with this platform is the therapeutic research. So we did a couple of custom electrode designs, providing them for clinicians also, which is different because there you have the full responsibility as medical device manufacturer, whereas here, I mean, all these regulatory, hurdles and stuff you outsource. We really here can focus on our competence and development and manufacturing, and let the other ones take care of regulatory approvals and marketing. And but okay, here we have to be responsible for the documentation, but for research, the hurdles are a little bit lower than for a full market approval. And especially here in the US, the FDA, with their early feasibility trials, it seems that the FDA is decreasing the hurdles for clinical research. And it seems to work because like 10 years ago, all the companies were going to Europe, doing their clinical trials there, getting market approval there because the regulatory hurdles were lower. Now it has changed, or at least Europe is as high when it comes to product approval, but the FDA makes it easier And I really have to say the communication I had with the FDA compared to the communication with the notified bodies is so much better. You might criticize the FDA. I don't know what experience they have, but I really like the FDA. I would say. But okay, here we have the responsibility. We need to have this fully developed and documented. But we want to use this platform to develop new clinical applications and new therapies, but not alone, but together with partners. So we want to provide it to clinicians like, for example, Nathan Rowland, who was presenting the dinner speech, and to help him do, for example, an innovative stroke rehabilitation or closed-loop Parkinson project. Or also industry. Or maybe do some own research if our funding permits it, depending on the money we have. But what we don't want to do is, go for approval for a new therapy and then market it, because then you really would need to specialize. We are now a company that's like mid-size. We are more than 60 people, but that's still small. And if you want to go for a new therapy, then you need to be an epilepsy company. You need to specialize in epilepsy, and that's it. And you have to go this, and it doesn't fit to this model. It also wouldn't fit to our customers who would fear competition. They would think like, why you're going for this? So no, I don't want to buy your technology. And in the end, it's very risky. So I tend to say I prefer to work on 20 new applications together with partners versus working on one alone. That's our model, how we go and how we leverage our platform. So the technology business that we are doing has essentially 3 phases. Typically, a customer comes with an idea to us. We help him discussing what you need to pay attention to, and then we build prototypes for him. That's typically going on within a few months. One can do some testing, and once the customer is happy and says, okay, now we are going into medical grade development. Depending on what he wants, what he needs, we can provide either full documentation, like do the biocompatibility testing for him, etc., or just deliver the devices to him that are then manufactured under design control and with the appropriate qualified processes. And then he does the testing. He can do clinical trials, market approval, and then after that, we would just manufacture and supply. We started marketing and sales, especially for our array electrode and some other components. So we had to work on our website, some product catalogs I put as an example there, and on the table behind we are going to exhibitions. On the one hand, that is the good old neuroscience and scientific meetings where we were coming from, and where people use our technology for their research, but also on the industry exhibitions. We did a marketing campaign here with this Maddie here, where we said, we called it as precious as your research, and we put our electrodes and have her wear them as a kind of jewelry or something to attract some attention. So yeah, that's things that are also fun and to work on that, but it's also work that needs to be done. But I think with the electrodes we are very well positioned on the market. And a lot of work obviously then went into the team. Team and organization is so critical, because in the end the team needs to do all this, and I cannot bring a single electrode out to a customer without involving at least typically 4 or 5 different departments. So from sales to discussions with engineers, then maybe checking by quality management, then it goes to the engineers, they design it, manufacturing, quality control, and then sending it out. And all this can only work if you establish good processes. When you're a company now of more than 60 people, you have to have processes in operation, because it's too large that everybody knows what's going on. And you have to work efficiently together, and so otherwise you lose money and time. So that's a lot of work going on. An important step was this Martin Schüttler. He was on the founder's picture from 2011, and then when we had these problems, and then I came and told him and the professor Thomas Stieglitz, I need one of you in the company. So Thomas Stieglitz didn't give up his professorship, but Martin gradually moved from being a part-time CTO sticking to university, getting a 2 years leave from his permanent position at university, to then finally quitting university and joining me as a CEO of the company. So his title here is CTO, but in fact we are both CEOs, and with like managing the company together, and he's responsible for the larger groups like development and manufacturing, and I take care of the rest. We have 4 development teams. There are now 8 people who are electrode engineers, and we have 4 in electronics and 6 in software, and then we have another 6 around in reliability engineering. They do the hermetic packaging, and they make sure that before manufacturing starts in the manufacturing team, that all the processes are reliable. And that's how we are now organized. We have a board here where our investors are represented, and some industry experts that is advising but also controlling us. We have a scientific advisory board. Everything is still relatively local. It has the advantage that we have short ways and everybody can come easily, but it's also I'm working on getting it a bit more international. With the team, it's already like we have maybe 20% or 25% non-Germans coming from all over the world, and for me it's important to have also the team more international, and I hope board and scientific board will follow. Last year we finally moved out of university. I mean, university was great. We had this qualified clean room already there. We had all the machines there. We didn't have to invest in this. We had offices, very, very crowded at the end. But it was very good to have this infrastructure, because otherwise we would have to build it for starting. So we could start right away doing all this, but in the end, as a medical device and implant manufacturer, sharing a clean room with students who are there in from time to time. I mean, even if like we were like probably 80% using this clean room and really heavily occupying it, we needed to go out at one point, and so we built our own clean room in a building that's newly being built. Now we have a 400 square meter clean room. So we have much more manufacturing capability. This is the new building where we are sitting on the complete ground floor on 1,500 square meters, still located in Freiburg. So if anybody of you is in Freiburg, I'll be happy to have you visiting us. There are a lot of other things also going on. So who knows what an ERP system is? One. Yeah. How do you know? Why do you know? Okay, you work in industry, yeah. So enterprise resource processing, or I don't even know, but it's the system that essentially is one system where all your goods are into it. So it's like connecting all your processes. So like when you buy something, it's immediately in this system. When you use any material, it's drawn from the stock managed by the system. It monitors everything. All the quotations are in the system, and we introduced this system because our investors thought it's a good idea to make us more professional. It's probably was a little bit too early, but and it's also still not completed. It's good to have it, but it costs a lot of time and energy and discipline. We introduced a project management in the company. Now we have a team of 4 people who are doing only project management, taking the resources from the different development departments and quality management. I had to complete 2 more financing rounds, and hopefully the third and probably final one will be completed in the next weeks. And I was lucky because I think I was lucky because I had always found private investors, which on the one hand are much more aligned with the business model we have, which is not focused on the one killer application, which is typically what venture capitalists like. They would say, yeah, it's nice you make some revenue with your electrode, but it's just distraction, from the big goal. Because if you make revenue, okay, it's nice, but it's not giving the company probably like $1 billion of value. On the other hand, the private investor sees, okay, they make some revenue, so it's at all anything good what they are doing, and in case like the big goals fail, I will not lose my money completely. I mean, the VC, they calculate with a, like 10% are successful, 20% will be maybe sold for just to get the money back, and the rest of the 70% will be gone. But private investors think different, and me as a founder too. I mean, I probably if I'd be in IT where I can try out for 2 years or so, I would say, okay, I'll go for the 10% big success chance, and then after the 2 years I do something else. But after, in medical device where things take so long, I wouldn't want to have a 10% success chance of the company. That would be not so nice for me. We also acquired additional grants all the time for German or European means quite successful, but compared to what is offered here in the US, it's very, very little, and sometimes also not so efficient. Because you need to work for the grant, you have a plan that's not working out, and you need to focus. So anything below $1 million or so in grant money, that is not also really very well aligned with your internal development pipeline, I would say don't do it. Just complicating. I'm almost coming to an end. That's a kind of the story how we tell it now and how it's seen through all the years. So we are located in Freiburg, that's in the southwest of Germany and kind of Central Europe next to France and Switzerland. Very nice area, little university town. Black Forest around for good hiking, and mountain biking has become a great action. There are a lot of trails there. A lot of my staff are going biking all the time. Maybe just some words to the challenges we are facing now. So one challenge is complementation of our team. In the end, it's the people working in the company that drive it, and to get experienced and talented staff is difficult. I mean, we are happy to sitting still next to the university in a university town, so we get good and motivated young personnel, but you also want to have some experienced personnel, and that's, I think, the same. As in Europe currently, it's really hard to get. We were just, for example, looking for an IT person like for a year or so, because there's just so thought, and I mean, we are not really a rich company, so we cannot say, I'll just pay, whatever they want. So we need to have like a more normal payment schedule. It also gets more and more transparent. So they want to have all the who earns what in Europe. They want to make it transparent. It's not completely there yet, but it's going into this direction. So that's a very important thing. We're always looking for new people. So if you want to join us, you can take a look there. yeah, internal organization is in a growing company. You know, all the time you have to adapt the processes, and communication is really, really key. It's, you know, that's something you need to culture and pay attention to, because if the people don't communicate, things will go wrong, things will be inefficient, and it's something you don't know from the start, and especially a certain type of maybe a male engineer who's, you know, totally clever, totally specialized, but he's not so fond of communicating. He thinks he knows how to do it. He just wants to do it. He goes ahead, does it, and then, you know, after like half a year, like, did you talk about this with the software person? Well, no, but this is clear, and yeah, we saw it briefly, and then it comes out, no, it was just done wrong. It was not paid attention to, and okay, you lost half a year again. And also, do you need to cultivate the people to speak out on problems? And I had a very clever technician, where we had the problems with a cleaning machine once, and in the end, he would tell me, well, I told you this already, or I knew this already a year ago that this wouldn't work, you know. Okay, yeah, maybe you told it once in a very low voice, but why didn't you speak up if you were so sure this wouldn't work, you know? Yeah, that's and that is really something one has to pay attention to cultivate the communication. And on the one hand, of course, if everybody's sitting only in meetings and communicating, that's also problematic. So the key is efficient and transparent communication. Yeah, there's one of my investors is an entrepreneur by himself, but, you know, from a very basic education coming. He's been working very hard, built a number of companies, and he has a formula in his company saying that success is equivalent to its education or intelligence, and then it's the power you bring in, and then it's communication. It's all a product, but communication is squared. Yeah, because it doesn't help if you work hard and if you're highly educated, if the communication isn't done well, then you'll not succeed. And especially in such complex and interdisciplinary projects. Okay, and then another challenge is, of course, there are a lot of requests for our technology. Should we do this or not? Is it really on our line? How much do we invest into further development and testing? Do we go to market too early or not? So in the end, it is always a limit of resources. You know, we are growing, but anyhow, at any time, the staff will say, it's too much, we need more resources, we have not enough, and we cannot do this, and in addition, and so on. So indeed, the resources are limited, but in a growing company, it will always be limited. By definition, there's always more work to do than you have capacity. So you need to make good decisions, because otherwise also your team will not understand what you're deciding and gets confused. Yeah. That's it essentially. That's a little cartoon for maybe my position currently. So yeah, between investors and staff, and all are yelling. No one is ever content typically, and but still, you know, staff is complaining, not enough resources, it's too much stress. You know, investors, you know, progress is too slow, but still, you need to be in a very good mood. You need to motivate both of them. You know, that's a real challenge when you are on the top of a company. Typically, the good news they are taken for granted. They do not arrive you. All the bad news are coming to you, all the problems. But your role is to be, you know, positive and motivate the team. And that's something you have to work on. It's not the best of my abilities, I have to say. Probably, but I'm working on it. Yeah, okay, that's the final slide. So that's our company vision is that we want to be a leading partner in the development of new therapies in this field. That's what we are working on, but being a partner and not the one doing it alone. And I think partnership is important to move forward in this complex field we are all working on. Thanks.
Thank you for your talk. So I'm trying to understand your technology-based business a little more, so the electrode thing. Are you making revenue right now? Like, are you selling?
Yeah.
Okay.
Yeah, that's... I mean, it's still like 2/3 of the team are working on internal development projects, mainly finishing this, which is mainly software and test samples for all kinds of specific tests now. But I just here. So yeah, and we are making revenue here. So we have industry and academic customers for whom it's electrodes, encapsulations, and one or two systems-based projects. But it's mostly electrodes, and more cuff electrodes. There's also a project where we're doing transcutaneous electrodes, strip or grid electrodes. So it's all kinds of versions. And
how much of the money you're using is coming from the revenue that you're making right now versus grants and investments?
Yeah. So obviously, I wouldn't probably need to do another financing round if we could already finance ourselves. But it's such that 1/3 of the team roughly is working in this type of revenue business. And if you would look only at this 1/3, that would be profitable or at least break even. So that's okay. But of course, if you have 2/3 of the team still working on internal development, that needs to be financed by investors and grants. And it used to be for a long time like 1/3 of each, 1/3 revenue, 1/3 grants, 1/3 investors' money. Now grants are getting less and less because it's just small grants and team is growing. Revenues are increasing now, but until we have the complete system online here and have so many people working in R&D, it's now that we also need quite some investors' money. Also, we are in the new building now with all our new infrastructure that also needs to be paid. It's alone the power for the clean room is like 5,000 a month. So and that's just the power for the clean room. So it's adding up. Yeah.
So in your talk, you mentioned about the closed loop paradigm for your technology. What do you think is the biggest challenge regarding, why it's not fully adopted? Is it because the clinicians don't like that, or the end users, or what's your thoughts about moving from
the
an
challenge for what stage?
for the adoption of the closed loop paradigm versus the open loop paradigm?
I think first now we are going into a phase of clinical research. We need to find out and fine-tune the parameters of the closed-loop therapies. And so at this stage, it's not so much about the final adoption of the market. So at this stage, it's driven by those research-oriented clinicians and research consortia who want to develop new therapies. But of course, if you look at NeuroPace, who have a closed-loop therapy in there, there are, and not only NeuroPace, also if you look at the open-loop DBS for Parkinson, medical implants are not fully adopted. So they are stuck in this early adopters phase. So also the open loop Parkinson therapy is a wonderful therapy. It works quite well, but it's not fully adopted. And the question is why, and would this also be the same in the future? And that's I can only guess, but it's easier to prescribe a pill. There are people who are afraid of implantation, and it's also physicians who are, I mean, if the general physicians are not suggesting this therapy and bringing the patient to the appropriate hospitals who have the power to do this, I don't know, it's complex. I'm a little bit optimistic that since the new generation is growing up already with a kind of external implant, the smartphone, that somehow the adoption of technology that is then also implanted because it's just easier and more efficient, that it will improve. But that's just a guess.
Okay, thank you.
Do you have a sense of what accounts for the difference in funding availability between Europe and the United States?
Well, I haven't been out trying to raise funding here. I just applied for 2 times for a grant from the US. So the funding rounds are bigger, the valuations are higher, but also the competition is higher in the US. That's also clear. But essentially, you have more a higher investment culture, more venture capital going on. I get a summary every week from the German stock market, where they summarize the venture capital deals over the world. And in the US, it's always an order of magnitude higher, especially in the life sciences. You will have like $10-50 million plus deals, whereas there'll be one probably in Europe. In Europe, I mean, and especially in Germany, a lot of investment is done by companies, family-owned companies. So that they are driving and they are doing well, but they are not being, the cultural innovation. There's these old traditions, so that there's no, German or European, Google or so, or these companies who also bring a new culture. And so that's what motivates me also to stay independent, and because you with a company you also build the culture of a company and try to grow this. So it's also by tradition it's not that active. They try to change this. We'll see.
Thank you.
I was wondering if you could tell us a little bit more about how you get your products into the hospitals in Europe. Specifically, we heard from Neuropace about the process of phoning insurance companies to try and get their devices covered by outweighing, the long-term cost of the disease. And I'm just wondering how that would change in Europe with a lot of our countries having national health services, government funded.
Yeah, in Europe, I mean, I would say it's pretty much a disaster because you have, like 20-something countries in Europe, and all have their own system, and they are probably working on harmonizing this somehow, but it's not yet. So I essentially know the German system, and it's complicated. So I'm pretty happy with this business model because. But it already starts here, with the therapeutic research. So in the US, you have a thing that's called IDE. And if you have it once, you can have a master file somewhere and things get easier. And in Europe, there's no such a thing like an IDE. You can get clearance for one trial. That's maybe similar then, but it's not a generic procedure. And then you have, the rules for getting approval are harmonized in Europe, but for reimbursement, not. And so I have not the strategy of getting into reimbursement at this stage. We have the FDA cleared electrodes, so they are an established product. That's not a new product. They are similar to the AdTech, PMT, etc. So they'll get reimbursed like the others. And if I get approval for them finally in Europe too, they'll also be reimbursed by a mechanism which I probably don't understand, but they'll be reimbursed. But if you come up with a new device, you have to prepare this and talk to them, and it's also complex. And I don't know whether it's easier than in the US or not. I don't
Yeah.
know.
I wonder if you could comment a little more on the inboarding versus outboarding of components. You talked about, so you separated your processing from your recording. So you have power transmission, and you talked about, well, you had algorithms that needed processing that would draw a lot of power, and so you put that outside the body.
Yeah.
And then, but I mean, on the extreme cases, external electrodes versus internal electrodes, and I think those are pretty easy to understand why you might need one versus another. But then, how about sort of some of the more intermediate components? Maybe you could talk a little more about that.
Which component do you mean? I mean,
well, I'm thinking in terms of anything from well, power usage, heat dissipation. Putting people probably would prefer to have everything implanted all at once, never see it again.
Yeah.
But then you can't replace things easily.
Yeah, I mean that's. Oh, the graphics is okay. The artificial intelligence should be here. Sorry, but the thing is that if you have a high channel device and powerful software, so it's something that is contradictory to battery power, right? So so either you stay low channel and simple on the algorithm side, then you can put it in a battery-driven device. But if you want to, I mean, the first version has 32 channels, but I mean, we will probably without multiplexing can go up to 128. And so you cannot record in good quality and stream this data out based on a battery. I mean, also. The argument is clear. Okay, you implant it once and then you forget about it. On the other hand, like the cochlear implant is a very successful implant, and you have this external device that powers it, and it doesn't seem to bother the people too much. I mean, I think it's also a way of being accustomed to having something that you wear. And I think here the smartphone generation that we are now growing up will have an easier time having some external device. They probably even wanted to have some way of control of what's happening in there and see it. But it is, of course, an argument you always hear, and there are many who say, no, finally for a treatment, you want to have something that's fully implanted. And that's maybe also fine. I just say we are now in the phase where you need to find out the treatment parameters. You need to understand how the networks interact and where you need to stimulate and when and where to record from. And this you can only do with a system that has many channels and with a powerful and flexible software. So I just say for the phase that I think we are coming to now or in, where you need to determine these new therapies, you need such a system. When you find out, okay, it's exactly here, I can stimulate, I need to stimulate with this algorithm, then you can again shrink this into a clever ASIC and a battery-driven device maybe. But you need to find out first, and for this you need more a system like this one, and maybe for some applications it will be fine to have something that is where you wear something that is controlling it. Does this answer your question?
I just wanted you to expound on it.
Yeah, but it's an important question, and I had a lot of discussions with this on this also with Tim Dennison, who led this program at Medtronic, and if you look to his slides and how he sees the field, I think we're pretty much on the same terms there.
One more. If there were one technology that you could sort of have a wish, like I wish I had a shorter pulse laser, or I wish I had electrodes that didn't cause tissue reaction, what one thing would you want that would really move you forward?
Oh, that's a lot. I mean, we certainly need this this implantable connector here. That's a key missing component because here that's hardwired. So the ECOG electrode here is hardwired to the system. That's not so good, but the point is that there is no higher channel implantable connector. We are working on one, so I wish that this project is successful and we had the resources, but okay, it's probably not the absolute revolution. I don't know. I mean, yeah, electrodes without tissue reaction would be great. It's a good point. I'm probably too much specialized on getting this done, but I'll think about this.
Any other? Oh.
As you might have picked up, we heard from Neuropace the other day, and a few of us have had conversations about Neuropace where their device is implanted in the cranium, so they actually have to like pull out part of the cranium to implant it. So with your device, is it approximately the same size as a cochlear implant would be, or how did you decide like where to put it, how big it could be, and what the risks are of having it stick out a little bit from the cranium?
Yeah, so it has the size of the early cochlear implants. So nowadays they are a bit smaller, but still from the same concept. And it was from the start pretty clear that we place it in the cranium behind the ear for, on the one hand, better signal quality, and on the other hand, to prevent breakages of wires running through here. And we could reuse, so to say, the concept of the cochlear implant. That is essentially it. And if you, I mean, all the companies who are working now on similar systems, be it Neuralink or Naya Therapeutics from Dan Risado, I think they also have the hub here behind in the skull and behind the ear plant. So seems to be choice. But in fact, we also have a project where it's implanted in the chest, and we are preparing for this too, and also in order to also make the system available for the peripheral nervous system or the spinal cord.
Thank you for this very frank and a little intimidating talk. About the challenges of translating this technology. We've heard a bit over the last couple, especially this week, about how industry or even medical professionals might want something from these people. Mm-hmm. I mean, you're talking about partnerships, but it's not clear to me exactly what that means. So what if this class of young people have a whiz-bang idea? How do they become your partner?
Yeah, I mean by partnership, I mean the therapeutic research, such that we are the partner providing the technology, including the necessary technical documentation for getting it into human, but the actual therapy and the idea on how to treat and doing the clinical trial would be on the side of the partner. Or if it's just a technology business, then it's the startup company who's developing a new therapy, where we would supply one part of the system. So now if someone comes up with a new idea, so the general question is it a clinical idea, like a specific, or a technology? I mean, technology is, of course, I mean, we have a lot of technology, and then that's something different, but I think we are talking about a treatment idea or a clinical idea. Yeah, exactly. Yeah. So the process is then that we discuss these ideas all the time, and for us it's of course the better the clearer the idea is, and the more know-how and specifics are there. But it varies a lot, between the routine from an established company. They know very specifically what they want, versus probably an academic who has an idea for a therapy, and then we can say, okay, for example, for a first clinical trial, be it an animal or human, we would provide you with the electrode you need. It would be hardwired, and you could then use it, I don't know, for some up to 30 days or acutely in a trial. So we would design the electrode with him or her, and develop and manufacture it, and then provide it. And since we are, of course, a small company, so hopefully there will be some funding for this. We are generally giving lower rates to academics, only half the R&D costs, because academics don't have as much funding as industry. And on the other side, also the advantage of working with academics is that the work is more transparent. It gets easier published, so that's also an advantage for us, where we can say we can lower the rates. And sometimes if there's a very exciting project and there's no funding, we might say, okay, we support this project, so if it's interesting for us too. But obviously we are not in a position where we have a lot of money to spend on that yet. This might hopefully change in the future, so that we can do a little bit more together with researchers.
Quickly follow that up. Is there a tension between publishing and patenting? Does that get in the way of publishing?
Well, I always tell my research collaborators that they should patent more. Essentially, the problem is that I think it's a bit better in the US, because you're a bit more oriented maybe to spinning something out or commercialization. But in Europe and Germany, generally, the researchers are not patenting enough. And this is, of course, for us not so nice, because, if there is a patent, it's for us much easier to try a commercialization and attract investors' funding. And we as a company, we don't really publish anymore anyhow, just for reasons of time. So we try to patent. We also patent with research partners. Often we say, come on. The researcher says, well, I don't have time. And then we say, come on, write us a page. Then we add something, and then we make an invention together. And this we did a couple of times, and it works. Yeah. Good. Yeah. Good, thanks.